This is a production of Cornell University.
Great, thank you. Yeah, so as you mentioned, I'm Brennan. I am a PhD student in the Larry Smart Lab, and I'm actually in Geneva. And so I'm going to be talking a little bit today about some of the work I've been doing on monoecious willows. Right. So in the Smart Lab, we work on shrub willows, which are the section Vetrix in the genus Salix. And these have many uses, but we're primarily interested in them for bioenergy usage. So they're grown in a short rotation coppice setting, so high density planting setting, and they're harvested every 3 years, and the wood chips can then be burned to generate heat, or also used as a feedstock for biofuel. So I specifically work on the species Salix purpurea as a model system for the shrub willows. Now this, as I mentioned, it's dioecious. It has a ZW sex termination system, which means that males are ZZ and females are ZW. So the W is only inherited through the female line. And then it has about a 2 to 3 year generation time. So here on the left, these are the female catkins, and it's also a close relative of the poplars, which are another important biomass crop in North America. So, as I mentioned, this is a dioecious crop, but we do have a single clonal accession that is actually monoecious, named 94003. So here's a picture of it. I don't know if you can see it, but it has male and female flowers on the same catkin. So catkins are mixed and have varying amounts of male and female flowers. So one of the first things we wanted to do is characterize how does this monoecious sort of vary across environments, and can we get plants that are all male or all female but are otherwise monoecious. So we had 9403 already replicated across three different environments through a previous diversity study. So they were in randomized block designs in Geneva, New York, at Clairol in Portland, New York, and then at West Virginia University in Morgantown. And so I looked at flowering in the field and measured percent maleness. So basically how much of the percentage of each catkin is male relative to female. And for the Geneva plants, we actually took a subset and forced them 2 months early in the greenhouse to see if that changed percent maleness. And so what we found is that doing that, bringing them early into the greenhouse, that did not affect the percent maleness. However, we were able to model it. So the percent maleness of catkin I on shoot J in plot K in field L. So we modeled the shoot, and then the shoot was in plot as random effects, and then the field that it was in as a fixed effect. And we found that all of these had a significant effect on the percent maleness. So this indicates that there are both macro environment and micro environment effects that are sort of influencing how many male or female flowers there are on a given catkin. And so this box plot here, this just shows these are ones forced in the greenhouse. These are Geneva field, Morgantown field, and Portland field. And the gold are when we measure percent maleness by catkin, and the red are when we measure percent maleness by the whole shoot. And now if you'll notice, there's not a lot of difference in the means between when we look at the whole shoot or when we look at the individual catkin. But we do see pretty substantial differences between sites, notably those grown in Geneva had a much higher percentage female than those grown in Portland or Morgantown. Also of interest, if you'll notice the range on these, we found entire catkins and indeed entire shoots that were all male or all female. So if you look at a plant and maybe only has one shoot of catkins, they might all be male, and you might misidentify a monoecious plant as male. And that would be really important moving forward. So I mentioned we have 94003. So in 2015, the Smart Lab decided to attempt to self this to generate an S1 family. And part of this was both for genetic mapping studies and also to try to uncover the genetic basis for monoecious. So what's very interesting is that we were able to generate an S1 family of over 400 progeny that were viable, which that alone is pretty interesting because this species is an obligate outcrosser because it's dioecious, but we were able to get pretty healthy S1 family. So we have 4 years now of flowering data from this family, indicated here. And from that, we found that initially it looked like the family was segregated for monoecious male and female. But the ratio was kind of confusing. We weren't really sure how to model this for any genetic mechanism. But when we look more closely at the data, we realize that, so for monoecious plants, right, you only need 1 year of data to confirm they're monoecious, right? If you see a male and a female flower on the same catkin, you know it's monoecious. Now for the males, many of these had 3 or 4 years of data consistently supporting that they were males. But for the females, most of these only had 1 year of data, and then the plants later died or were very small. So we think that these females are actually monoecious and that we just didn't have enough data, enough catkins to really see any male flowers. Because as I mentioned on the previous slide, you can see all male or all female plants if you only have a few catkins. So in 2018, we took a single monoecious individual within this family and selfed it again to generate an S2 family. And now we've collected 3 years of flowering data on this family. And so once again, we see this confirms it appears to be segregating for monoecious and male. We only see a few females here, and these are likely false positives. They're likely monoecious. And so the ratio here is about 2 to 1, monoecious to male. And we collected GBS data on every individual in this S2 family. And then when we look at the sex determination locus, which is on chromosome 15, so that's what's determining sex. I remember males are ZZ, females are ZW. And when we look at that, we saw that that family appeared to be segregating for homozygous major allele and heterozygotes, but no homozygous minor allele genotypes, suggesting that it's segregating for ZW and ZZ, but there are no WW individuals, which, right, if we're crossing a ZW with a ZW, that's what we would expect. And then when we look at the corresponding sexes, the males seem to correspond to ZZ and the monoecious to ZW. So this led us to hypothesize that monoecious plants are ZW genotypically, and that there might be a modification on the W chromosome that's causing them to be monoecious instead of female. And that model is shown here, right? So we're calling this modified W, WH. So when we self our 94003, our monoecious plant, we get 25% male, 50% monoecious ZWH, and then the 25% WH, we think are lethal, because we don't see any of those appearing in our S2 family when we do the GBS data. So now to confirm this, though, I decided to cross this monoecious plant, outcross it to our reference male and our reference female genome. So we have a reference male and a reference female, for which we have fully assembled, fully annotated, high-quality genomes. So that's really facilitates downstream genetic studies. So when we outcrossed it to our reference male, Fish Creek, which this is ZZ, we saw about 50% male and 50% monoecious segregation in that family, which is what we'd expect if this monoecious mutation is in fact carried on 15W. Now you'll notice this is a little bit skewed towards male. Again, we only have 2 years of data on this family, and I'm almost certain that some of these males are actually monoecious, right? We only have 1 year of data, and I think as we get more years of data, it'll get closer to 50-50. But just with 2 years of data, we already see it's pretty close to half male, half monoecious, and really no females. I think it's really interesting when we cross this to our reference female, 94006. So we see about a quarter male, a quarter monoecious, but then about half of the family is actually female, which is way more than we'd expect otherwise. So we think, right, so 25% male ZZ, 25% female ZW, and 25% monoecious ZWH. And then we think this other 25% are WH, and that they're female. So this suggests to us that, okay, if a plant is monoecious for this modified W, it's lethal. But it might be that if it has a regular W and this modified WH, then that is viable and it's in fact female. So from that, we hypothesize that it's probably a large indel or inversion on the 15W, such that it results in a loss of function. And if you're homozygous for that, it's lethal. So this is just to summarize some of our hypothesis in our model based on these families. Right? So we think the causal locus for monoecious is on a modified 15W, which we're calling 15WH. Such that ZZ individuals are male, ZWH individuals are monoecious. If you're homozygous for WH, that's lethal, but a regular W and a modified W is lethal. So now moving on to the third part of this study is, right, so this is all interesting, all right, monoecious willows, but why do we really care about having monoecious willows? And so one of the original reasons we created this family was to perform genetic studies. So right now, these plants are only in single plant plots. We're in the process of scaling them up, but willow has a long generation time, so that takes a while. So even in single plant plots, we decided to collect data from high heritability traits, so notably SPAD and leaf dimensions. And these are traits that they've been shown to be high heritability, and they're also correlated with biomass yields, which is important for willow growth. So, and we were able to get actually quite a few QTLs just doing QTL mapping using single plant plots, which is very promising. So for most of the traits, so I only have 3 shown here, but we put up leaf area, leaf width, leaf shape, but all these leaf dimension traits are pretty correlated as one would expect and actually give very similar QTL profiles. So I only have leaf area shown here. But for most of these traits, we saw a very strong peak on 15W, which suggests that there might be some relationship with sex. So that was then included as a covariate, so we could actually pick out QTLs on other locations in the genome. Interestingly, when we did this for sex, we actually got a secondary QTL on chromosome 7, which was not expected at all. And so we're really interested in this focus as it might be playing a role in Monoecious. And in fact, there is actually a pedala 3 MADS gene in this confidence interval, which has been shown to be involved in male floral development and stamen development in Arabidopsis. Now for leaf area, we have quite a few QTLs that pop up, and for all the other leaf dimension traits as well. And many of these QTL have very promising candidate genes in them. Notably, in chromosome 16, there's a PIN1 homolog, and in chromosome 17, there's a Canadi homolog. And these 2 genes actually interact to specify the adaxial identity in leaf morphology. So that's a very promising candidate gene. And there's also a few other genes throughout these other QTL involved in leaf morphology and development as well. And then for STAD, we found multiple photosystem genes and thyroidoxin genes in these confidence intervals. So now, sort of the next steps of the study, right? We'd like to do a few more mapping studies using single plant plots of the S2 family, notably insect resistance and rust resistance, since these are also pretty high heritability, but we haven't had very good insect or rust incidents the past couple of years. This spring, we planned an expanded genetics trial, including replicates of this S2 family. And so in the next few years, we'll be able to use that to do QTL mapping of much more complex traits and slightly lower heritability traits. That'll be very interesting, especially since using only single plant plots, we already saw QTL for high heritability traits. And then we're in the process of resequencing a 94003, that Monoecious parent, and a select subset of the progeny from each of those families. So we've collected PacBio data from 9403, the parent, and we're in the process of assembling that into a new genome, which we can then, at that point, we'll hopefully have a Monoecious reference along with our male and female references. And then we're doing Illumina sequencing of some of the progeny, which will then map back to each of those references, which are the 3 different parents in those families. And that'll hopefully help us narrow down in on the actual locus that's causal for Monoecious. And with that, I'd like to thank the current and former members of the Smart Lab who have contributed to this research, as well as our funding, which has come from over the years from the USDA and UFA, as well as the NSF. And so with that, I'll take any questions if people have them.
Yeah, got a question. Yeah, so for this trait variation in natural populations for Monoecious, do you have a sense of, like, is there an adaptive trade-off going on in response to the environment?
Yeah, so the question was with Monoecious, is there an adaptive trade-off in the environment going on? So, it's thought that one of the reasons plants become dioecious is there's a disadvantage for if you're a male in dioecious, right? A male can't produce seeds, right? But it still uses resources. So for a Monoecious plant, right? We're not sure what exactly the adaptive trade-off would be. I mean, an advantage is, right, a Monoecious plant could colonize a new area, for example, right? It does, especially since it is self-fertile. Again, Monoecious willows are pretty rare, but they are observed, so we're not entirely sure. One thing in terms of the variation of male and female, those 2 sites that we saw higher percentage male were a bit in the previous summer when flower set actually occurs. They were a lot drier and warmer. And that with other species has been shown in other Monoecious crops to promote more male flowers. So that could be one reason we saw more male flowers in those other environments. Yeah.
Can you talk a little more about the indel that you mentioned on chromosome 15? I wonder how many genes the indel covers, or how long the indel is. And then also in the QTL that you showed, I didn't see a peak for the indel. Is that because you use SNPs, or because like the genetic analysis just didn't cover or include the phenotype?
Right. Okay. So yeah, those two questions. So the first one was, can I tell you more about the indel on 15? So that indel is hypothetical. I should have clarified that. So, we hypothesize that there's an indel. That's what we're hoping the sequencing information will actually confirm for us, right? And again, we're hypothesizing that because from these segregation ratios, it appears that 15W is responsible for Monoecious. It's somewhere on that chromosome. Otherwise, we wouldn't be seeing these ratios the way they are. And, furthermore, the fact that we get 50% female in this when we cross it to the reference female instead of a third of each, right, suggests that WWH is likely viable. And if that were the case, that means something's going on WH to make it lethal. And the most likely scenario for that would be a large indel, right? A single SNP might do that, but probably not. So that's why we're hypothesizing it's an indel. We don't know that yet, but the sequencing data we're collecting should confirm that pretty easily. If it's indel, that'll appear. So the second question then was about the QTL maps. You don't see a peak here at the 15. So there were peaks at 15 for all these traits, very strong peaks, like log scores of like 15, right? So that made the other peaks. When you have a strong peak, other QTLs tend to not be called or not show as high significance. And so I included that as a covariate so we could parse out these other QTL. Because the chromosome 15 is characterized by a very strong linkage disequilibrium. So it's very strongly linked. So it's hard to parse out any specific genes in there because everything is in strong linkage. So that's why we masked that. But yes, the QTL for sex without any covariates had a very, very strong peak on 15, which of course we expect because that's where sex is determined. Any more questions?
One other question. So you're like, I don't know much about monoecious plants or dioecious. Is that normal that this is really just a single locus, like sex determination, or could there be another locus?
Yeah, so the question was, is it normal that in monoecious and dioecious plants there's a single locus that determines sex? Yes, that is normal. So this is a binary trait, right? It's not exactly Mendelian, but it's a binary trait. It's not a quantitative trait. So yes, and in all examples of plants, as far as I know, there's only a single locus responsible for sex determination. And usually it's one or two genes, right? So in some cases it's a single gene that is a switch to male and female, and in some cases it's two genes. So one gene that might suppress male and one gene that might promote femaleness in the ZW system, for example, but linked together such that there's never any crossover between them.
All right, well,
thank you.
Well, let's thank Brendan. Yeah.
This has been a production of Cornell University, on the web at cornell.edu.